Thank you.
Thank you so much.
All right. I hope all of you can hear me. So let's just go ahead.
Yeah.
Can you see the slides?
Yes, that's your slide.
So I want to talk about the distinct secretomes of P6 and P21 positive senescent cells across multiple tissues. I had the pleasure to be in the COSA lab for 2 years and continue this analysis that I'm just talking about today. So just a brief overview, and I'm sure all of you are familiar with this. The concept of cellular senescence was established a couple of years ago, and it has been shown in baboons that a lot of telomere damage is accumulated throughout the lifespan. This was the first thing that has been discovered. And so in a normal cell, just with various stimuli, it can be DNA damage, it can be exhaustion or oxidative stress or any inflammation, might just undergo apoptosis and everything is fine. But it might also accumulate some factors like P16 or P21 and then become a senescent cell that damages the surrounding tissues with a senescence-associated secretory phenotype, or called SASP, that you can see over here. That again in turn has other effects on these tissues. For instance, it might affect wound healing. It definitely affects bone healing, tissue remodeling, and so forth. So the two questions we were asking is, what is this SASP? Can we define it a little further? And the second question was, is it different in P6 and P21 positive cells? And what kind of the SASP is probably just P16 or just P21 associated? And is there maybe some common ground for both of them? So first, what we did is that we kind of did a meta-analysis. We looked through a lot of studies and just included the studies that experimentally validated a couple of senescence-associated secretory phenotype markers, so the SASP marker. And with these studies, we found 125 genes that are closely connected, and looked at if these genes in a form together in a gene set can show us that there's an aging process. And we compared young and old samples for the first thing in bone alone, because we're primarily a bone lab. And we found, well, this gene set seems to distinguish very nicely between young and old samples. Then we looked a step further and looked in the brain, which is totally different tissue in many regards, and also found that the SenMayo, this gene set that we just created, was very nicely able to distinguish between young and old samples. And then we were able to publish this in Nature Communications. And the SASP itself consists of a lot of different factors that can be subsumized into cytokines, growth factors, modulators. And these are the 125 genes called as SenMayo that we were able to identify. And these form a very dense network that is closely connected. If you look at what exactly these factors are and what gene sets are enriched in this, interleukin signaling is one of the pathways that's highly upregulated, and cytokine signaling, chemokine signaling, and so forth. So this is the SASP that we were able to identify based on the literature, and that we could then confirm works to differentiate between young and old samples, and shows what these aging cells secrete throughout the lifespan. So in this project that then followed as logical consequence of this, we wanted to know what happens in P16 or P21 positive cells. And we started with a brain sample. Over here you see the different cell types we got in the brain. And if we just originally look at all these cell types in percentage, then you can find this bar over there. But we knew that the senescent cells themselves needed some criteria to so we needed some criteria to narrow it down to cells that might be senescent in these single cell RNA-seq datasets. First thing, we wanted just to include CD45 negative to get rid of the hematopoietic cells, because we don't really believe that there's a strong senescent phenotype in these hematopoietic cells. There might be one, but we just want to focus on the non-hematopoietic cells over here. The next thing, we know that these senescent cells needed to be Ki67 negative, so not replicative anymore, which again changes the balance of cells that are left. And finally, we wanted to just exclude cells that are in the S phase, because we knew these could not be senescent. And from this selected population, we then went ahead and just chose the cells that are either P16 positive on the top here, or P21 here on the bottom. And we wanted to further define these different cell types. And this is just the population that you saw before, but now just marked as red cells are p16 positive cells, the blue ones are p21, and there are very few double positive cells that are both p16 and p21 positive. In this dataset, it's just 3 cells. So, and we made sure that the markers we selected were correct. And then we selected the SenMayo and looked which of these factors are secreted by the cells that are p16 positive, which are shown in red here, or p21 positive, which are shown in blue over there. You see a lot of factors are secreted by p21 positive cells, not so much by p16 positive cells. And there's very few overlap. In this example, it's CXCL16 and PLOR that are secreted by both of these different cell types. So keep this pattern in mind because it will follow us through all the tissues that we're going to have a look like. So this is the brain and applied to SenMayo. Next thing we did was that we use a different gene list because someone might ask the question, well, maybe SenMayo is just biased towards p21 positive cells, which is why you see so many factors coming from there. But we used a different gene set that just focuses on all secreted factors that were described and experimentally validated, which are altogether like 900 something genes. And the pattern is still very similar, but now we got a lot more secreted factors for the p21 positive cells, a lot more from p16, but still more in the p21 population. And there are very tiny overlap between these two populations with the blue genes over there. So this is the example I had from brain, and I won't take that much time for the other tissues that we analyzed. This is the muscle tissue single-cell RNA-seq data set that we used. And again, we were able to use the selection criteria from before, so CD45 negativity, Ki67 negativity, S-phase negativity, and then select the p16 or p21 positive cells. And again, you find very few double positive cells. One of them is, for instance, over here in the endothelial cell cluster, but altogether there are very few double positive cells. And again, you were able to see this pattern coming from SenMayo that is secreted by p21 positive cells, a huge amount of SASP factors that is secreted by these cells, whereas the p16 positive cells just have very few factors that are secreted just by them. And then again, some overlap between both of these. This is the pattern that you see when you look at the whole secretome that has been described. Sorry, you want to ask a question now, or yeah, maybe now? Because you've raised your hand.
Dominik, just curious to see, are you using the SASP module from the SenMayo, or you are looking for a more broader gene list of SenMayo, SASP?
Yeah, so the first one is just SenMayo, and the genes that we were able to identify and that have been experimentally validated. This is the gene list that is just described as the whole secretome from cells that has been described in literature from humans. So this might be a little more biased, but we wanted to confirm that the pattern we see can also be seen from these different gene lists and not just from the SenMayo, because some might argue this is more towards p6 and p21. So we wanted to have a completely different gene set, the whole secretome, which is what you see here.
I see. And another question is that in any of the red or yellow blue dot here, these are the genes expression detected from the corresponding cells, right?
Right. And the size of the circles, sorry, I should have mentioned that, is the fold change compared to all the others. So the ones that are largest are the ones that have the highest fold change compared to all other cells.
Thank you.
Right. So
Hi,
then
Dominik.
Oh, sorry. Yeah, go ahead.
May I have a question regarding that p21 positive cells?
Yeah.
Because in skeletal muscle, a lot of the mononuclear cells are p21 positive, but they are not senescent. So
Yeah.
is it better to set up a threshold for p21 high expression versus low expression?
Yeah.
Otherwise, here there are just a lot of normal cells, I guess.
Very good idea, but of course not all p21 positive are senescent, but as I said before, we just included the CD45 negative, Ki67 negative, and then S negative cells. But again, I don't think that all of these cells, or that we capture all senescent, well, we capture all senescent cells, but maybe not all of these are senescent. But if you start with a threshold for the muscle, then comes to the question, what would be the appropriate threshold? I mean, then we would need to ask the same question for the brain, bone, and all the other samples. So yeah, sure, it would be great to use that, but I wouldn't be sure how to do that reliably, because can you for sure say that in muscle there are 10% senescent cells, no matter the age of the organism or the sample or the I don't know.
Yeah, I understand your point, but my concern is in skeletal muscle, based on our understanding, these mononuclear cells, regardless of P29 expression, they probably are not senescent.
Yeah.
Yeah.
It might be that we don't just capture or that we capture cells that are not senescent and necessarily senescent. This is the closest we can get without getting too much bias towards a threshold. And as soon as a valid experimental validation of a threshold for each organ of the amount of senescent cells, they're not only fine with adjusting all of these results for each organ. And I think still the threshold will be different for brain compared to muscle or to bone, or even injured muscle or bone. But I didn't want to bias one of the organs too much, so we chose not to use a threshold for all of them. But
another point I want to make is we found in skeletal muscle the P29 positive senescent cells may be the myofibers, which were not included in the single cell seq experiment. We have another data set that's using a single myofiber RNA-seq that you could probably look at.
You said the myofibers are more senescent cells than in this one that you are obviously familiar with?
And I would say that's maybe more like P29 related.
Okay. Yeah, sure. Why not? We might include this one in parallel for future revisions. So, because as you see in the next one, we choose to parallel some organs anyway.
Yeah, we can further discuss the details.
Maybe you can send me the paper and we can. Perfect. Okay, so the next organ, because as I said, we're a bone lab originally, was the bone sample, and we used the same approach, SenRNA-seq, then the previously mentioned selection criteria, and then the P16 or P21 positive cells. And again, the pattern stays consistent. You got a lot of secrete factors from P21 positive cells, not so much from P16, and just very few overlap, which might be interesting that it's unit as PP1. But in this case, we were able, because Madison Doolittle published another paper in Nature Communications and had practically another bone sample, but now used the CyTOF, so a proteomics approach. And with this one, we were able to also validate which cells are P16 or P21 positive. But the caveat here is that we just had a certain amount of genes, because CyTOF uses these antibodies, and we just had 32 genes or proteins that we could have a look at. And this is why this pattern looks a little different. We were still able to see that some genes like IL-6 are typical for P21, others are typical for P16 positive cells. But since we just had 32 genes, the final findings are a little different from all the others, I guess. But still, we were able to include the proteomics analysis from bone over here. This would be for the single cell RNA-seq and the whole secretome. And again, you see a lot of P21 SASP secreted, not so much from P16, but a huge overlap over here in the bone. The next data set we used was a liver sample. And again, P21, P16, and a few double positive cells. You see a lot of P16 secretome, not so much P16, very few overlap. And now we chose to, so sorry, this is the whole secretome. Again, you know the pattern, P21 a lot, P16 not so much. Now, since all of the previous organs have been mouse organs, we chose to additionally analyze human samples. So the human skin was one of the tissues we used, and we found a lot of P21 positivity, not so much P16 and not so much overlap. And in the skin, we could not find a single gene that was secreted by P21 and P16 positive cells. All of them were very distinct, which is the first data set we found this. So to make sure this is not just a species effect, we also included another data set. So here's the human skin whole secretome. Now we got a little overlap between both of these, but still most of it is distinct. Then we used the human lung data set and found P16, P21, and very few double positive cells. And again, a lot of P21 secretome, not so much P16, very tiny overlap. Question?
Yeah, so Dominik, I have a question about the double positive cells. So
Yeah.
does those cells tend to be secreted, like those more of those yellow shared secretomes or not? And have you guys used the doublet finder to confirm that this double positive cells is not two cells profiled together, but really is one single cell?
Yes, we did to check whether these are just doublets. They're not. And regarding the secretome, it was all of these, so both the P21 and the P16 and the yellow ones, not exclusively the yellow ones. But these cells are so few. In some datasets, we have like from 60,000 cells, just 4 or 10 that are double positive. So I'm not sure this is really the truth, the biological truth that we see here, or just a technical difficulty, because so few cells, even the mathematics behind it to find the cells, the secretary pattern is heavily biased. So in the paper, we don't focus on these because I don't really believe in that there are a lot of cells that are really double positive, which is why I think P16 is very distinct from P21 positivity. Yeah, good point. Thank you.
And all these different like red, blue, green dots, their definition for P16 or P21 positive are based on whether they express the cells, express the genes at all or not, right? So this is
so they're not, they don't have zero. They don't have zeros. They're more than, they have some expression that's more than 0. So
Okay.
0 is excluded. That's right. It's not just that they don't have nothing, because that wouldn't make sense, but more than 0 was the threshold.
Okay.
And as Joe mentioned, we chose not to use a specific threshold for all the tissues, but above 0 would be positivity, right? From the scaled expression, not from the rare expression.
Thank you.
Yeah. Okay, so the whole secretome for the human lung still looks P21, has a lot, P16 not so much, very few overlap. So to summarize all of this, we compared the P16 and P21 associated SASP throughout all the tissues. And again, the circle size marks the log2-fold change. And for the whole tissues that you can see color-coded and the P16 positive cells, you find these markers that are just from SenMayo. And these markers that are just from SenMayo, but for the P21 positive cells. And you find, interestingly, the lung has the highest log2-fold change, but just for the P21 positive cells, which is strange, but seems to be the truth since it's not the same for P16. So not a technical problem over here, but a more diverse and much broader secretome that we find in the P21 compared to the P16 positive cells, which is interesting by itself, but it underpins the idea that these cell types are indeed really distinct from each other. And another way to look at these and just to focus on specific factors and data sets that we found these factors to be associated with, when we just look at the P16 positive cells and then look at in how many data sets were these specific markers enriched that we find in P16 positive cells. In 3 of the analyzed data sets, we found SPP1 to be upregulated in P16 positive cells. So this really seems to be a marker that is closely connected to P16 positivity. Whereas in the P21 positive cells, we find 2 markers that are enriched in every P21 positive, or in the majority of P21 positive cells, in 6 out of the 6 analyzed data sets. So UN and ICAM1 are the markers that really seem to be associated with P21. And there are some markers, if we combine both of these, that might be something like a core SASP. So that seems to be for each, if you want to call them senescent cells, markers associated with the secretome. So UN and IGF-BP4 would be 2 of them. This is just from SenMayo. Now, if we again want to broaden the focus here, so you have in mind Yun is the most important one. And then we use the whole secretome, and we find more markers now, but the maximum data sets that we find P16 positive cells to secrete are 3. So there's never 6 out of 6 data sets in which P16 positive cells secrete a specific factor. It's a maximum of 3. So you see the SASP seems to be smaller and not throughout all the tissues that we can find in P21 positive cells. So these are the markers, the whole secretome markers for P16. And then the P21 positive cells, we again find Yun that is secreted in 6 out of 6 data sets, as we were expecting, because we knew that from SenMayo. But there are a few others that are at least secreted in 5 different data sets, like A3 or EDM1. Then the core SASP that might be secreted by both of these cell types is just defined as a sum of the P16 and P21 positive cells, and then secreting. It's not surprising that Yun is the most important one, because 6 of the 6 data sets are from P21 positive cells, and just one P16 positive cell type secretes this Yun. The second one, which is secreted by 4 out of 6 from the P21 and 3 out of 6 from the P16 positive cell data set, is Spark, which is interesting because we know this factor from the bone field anyway. So these seem to be the markers that are something that comes closest to a core SASP that might be independent of P16 or P21. And next step we were doing then to further validate that P16 and P21 positive is indeed very distinct is that, and I will just show this briefly for the brain. And we looked at all these different cell types, and then we looked at the signaling pattern that they were secreting to the other cells. But this time we included P16 and P21 positive cells as an own entity to see what these cells secrete. And for instance, you see the P21 positive cells were secreting, so these are the senders over here, P16 positive cells, and they secrete to these cells on top there. So one pathway they use is that they secrete CCR4, and they secreted to these CCR5 receptors that are mainly from the microglia, which is color-coded in blue, as you see over here. And for the P16 positive cells, for instance, you see that these prefer to secrete via GRN, and they secrete both to the neurons over there, to the SORT1 receptor, and to the SORT1 receptor of oligodendrocytes. So this is just a general pattern we were following throughout all the tissues, but exemplarily shown in the brain. Just we introduce these P21 and P16 positive populations over here. And now you can combine all of these and see whether these are indeed distinct and follow distinct secretory patterns throughout all the tissues. So for instance, this is a brain, and an easy way to depict this is a heat map. So continuously let this heat map grow. You find that P21 and P16 positive cells secrete via these pathways. And you can measure the secretory activity via this relative centrality strength. And in blue is very low, and in red is very high. So, and you find that you can also do this nice Sankey plots over here. These are the pathways that we had to look at. And the P16 positive cells, for instance, secrete via APP pathway to these oligodendrocytes. So one difference we find over here is that we see that the MAC pathway is not so much used on the P21 positive cells, so which is why it's blue. P21 doesn't secrete a lot to the oligodendrocytes via MAC pathway, but P16 positive cells secrete a lot, which is why it's red, to these oligodendrocytes over here. So this is how this heat map is built. The point I want to make over here is that there are specific differences between P21 and P16 positive cells. In this case, it's primarily the MAC pathway, which underpins the statement that these cells are indeed distinct from each other. In the muscle, we had too few P16 positive cells to compare these both together, which is why we just have P21 positivity and the secretome shown over here. In the bone by itself, we found that 2 pathways were interesting to have a look at, THBS and FN1. And you can see P21 positive cells secrete a lot via these pathways to hypertrophic chondrocytes and to this osteolineage cell type 2, which is why these are red. And this doesn't happen in P16 positive cells, which is why it's so pale over here. In the liver sample, we see some pathways are very similar to each other for these two cell types, but for instance, YAM is used a lot in P21, but not so much in P16 positive cells. These were the urine tissues, and the human skin had these CD99 and desmosome pathways that are highly different. So desmosome pathway is not used that much from P16, but a lot from P21 positive cells, mostly to secrete to granulocytes and spinal cells. And in the lung tissue, finally, we see a lot of differences. One that is very interesting is FN1, because P16 positive cells seem to secrete a lot via FN1, but P21 cells don't do that at all. So another way to show that these cell types are distinct from each other because they behave differently regarding their secretory activity across all these tissues and even across species. To do the next step, the next level up from the secretory activity, but now look at the controllers for all these cell types and P16 and P21 across tissues. We had a look at the regulatory activity across these tissues just for P16 positive cells, which you can see over here. And we just included the regulons for these P16 positive cells across tissues that are highly relevant because of their relative activity above 1, which is a parameter to just show how important these regulators are. And then we compare this to the P21 relative activity, again, across tissues that we had, and we could barely find any regulators that were the same across the tissues and between P16 and P21, which is interesting because it again states that these cell types are completely different from each other, and it indicates why we don't have a lot of double positive cells, because these cell types are so much different from each other. Okay, to finally briefly sum this up, we know that senescent cells rise with aging and are characterized by either P16 or P21 positivity, and I don't believe that there's a lot of double positive cells who are really double positive, at least from the single RNA-seq and site-of data that I could get my hands on. We know that SenMayo characterizes the SASP reliably across tissues and species, and we use selection criteria, CD45 negativity, KS67 negativity, S negativity to identify presumably senescent cells. You're right, Zhu, maybe we don't re-identify too much of them, but I think in this case it's better to have too much instead of too little. Then from there we select P16 and P21 positive cells, and we very rarely find double positive cells across tissues. We found that the SASP varies depending on the organ and whether it's P16 or P21 positivity that we followed up on. We found that P16 associated SASP, P21 associated SASP, then that these were organ dependent, and we also find a core SASP that might be needed to for every senescent or pre-senescent or senescence-like cell to secrete to influence the neighboring organs and cells. We found that the most important core factors are UN and SPARC, and we saw using SenNet that the communicational pattern of P16 and P21 cells varies a lot across tissues, and that these several core regulants controlling P16 or P21 positive cells were different between organs. So just the two headlines you might memorize from this talk is that there's a diversity of stellar senescence and SASP across tissues, and that it might be very interesting in the future to highlight the differences between both of them for more nuanced investigation between P16 and P21 positive cells. Some of them might be more or less injury or senescence associated, and the SASPs that expresses this might just be connected to these cell types. Okay. If you're interested in the preprint that you mentioned already, we put a bioarchives, and you can just follow this QR code. And thank you for your attention. Yeah, that's it. That's a beautiful Mayo clinic. I had the pleasure to work for 2 years.